This is the mini lecture for protein structure. So you've been exposed to one of the major themes of biology, the relationship between structure and function. In biology, we say that form follows function, and that means that the structure of an object reflects the job of the object. One of the best examples of this is proteins. When I hear the word protein, the first thing that usually pops into my mind is food, meat, dairy products, eggs, that sort of thing. But that's because I eat constantly. However, even when my appetite is saying salad, I always make sure that my salad has a good amount of protein in the form of nuts, seeds, beans. Proteins in food, they get broken down into their components called amino acids. And then your body recycles these amino acids by making proteins of your own, like one big recycling plant. All enzymes are proteins, but not all proteins are enzymes. Enzymes basically allow the catabolism and anabolism that proceeds at a pace that supports life. We've got proteins that function in transport, like hemoglobin in our red blood cells. We have antibodies that cover invaders, marking it for destruction by other cells. We use proteins as hormones in our bodies, like insulin and epinephrine. I know, this is the same picture, but proteins, they get inserted into cell membranes to communicate with outside agents. Looking at the human body macroscopically, we've got contractile proteins in muscles, structural proteins like collagen and keratin in our hair, skin, and nails, and finally, proteins function in energy storage like seeds, nuts, and eggs. And this is why it's important to have these things in your diet. When I think of protein structure like those, I think of those like folded paper fortune tellers that we made in second grade. What's your favorite color? What's your favorite number? And then you open up the fortune teller and it tells you who you're going to marry, right? I married Joe, Mr. Sherman. So despite your potential spouse, the fortune teller, it's an important example because since it's folded specifically, it creates a very specific final product or final shape. In fact, there are different levels of folding that are used to create the final products of proteins. They're similar in that they're folded 4 different times to create a final structure. The primary level of protein structure is simply the linear sequence of amino acids that are strung together, and they make a protein chain like the one down at the bottom of the screen here. These amino acids, they're connected by peptide bonds, and so this is called a polypeptide. Every protein has a unique amino acid sequence, which is determined by a DNA sequence. There are only 20 amino acids that exist on Earth. Remember, that we know of, because science is limited by what's currently known. So a huge variety of these proteins can be constructed from 20 different amino acids. They contribute to this vast biodiversity on Earth. Some of these amino acids your body can assemble, but some you have to be sure to obtain in your diet. If you eat other animals, you're good, you're getting them all. If you eat only plants, you just have to take care to obtain the essential and conditionally essential amino acids. Corns, beans, and spinach, people, they got them all. The secondary level of protein creates 2 common structures, and we can see them here. We've got an alpha helix, which is a region of a protein that's twisted into a tight spiral. The coils of the helix are held together by hydrogen bonds. Other regions of protein, they fold up like a paper fan. This type of structure is referred to as a beta-pleated sheet. The alpha helices and the beta-pleated sheets together, they comprise the secondary structure of a protein. So a given protein, it might have 3 alpha helices and 4 beta-pleated sheets, while another has 8 and 2, and so on. Most proteins, they have at least some of each alpha and beta. Once all the helices and sheets are folded, coiled, and locked into place, the polypeptide takes on this like distinct three-dimensional shape. And this is the third or tertiary level of protein here that's indicated by a blob. But that's the tertiary level is kind of like the overall three-dimensional shape. We can go a little bit further though. We have the fourth level or quaternary level of protein structure, which describes interactions among different polypeptides. Not all proteins have a quaternary structure, but the big ones usually do. Some complex proteins are composed of more than one polypeptide chain that bond together to form a super protein. For example, the blood protein hemoglobin is composed of 4 different polypeptide chains and they interlock together. They have a hema pigment in it, which picks up the oxygen. In sickle cell anemia, it's a genetic disease. The DNA that dictates the amino acid sequence of hemoglobin is mutated, and it dictates an amino acid sequence that does not fold together like the one here on the right. This limits the amount of oxygen one hemoglobin molecule can carry. But it also affects the shape of the red blood cells, making them sickle instead of bioconcave, which is essentially round. Yes, sickle cell sucks, but someone with it actually has a less chance of contracting malaria than someone with bioconcave red blood cells. So I guess if you have sickle cell anemia, you have an advantage in southern climates. We see how the protein structure is made through these levels of folding, but we can also deconstruct a protein. This process of changing the protein shape is called denaturing. Denaturing usually involves a chemical or heat. I love me some eggs scrambled. When I cook the eggs, I'm denaturing the storage proteins in the egg. You are denaturing meat when you add a marinade, especially an acidic one. We can actually denature proteins, making them safe for consumption without using heat. Fish exposed to acids is called sous vide and is safe to eat. So are eggs that are exposed to lemon juice. In fact, a good sour mix can be used in many sweet drinks, perfect for a summer day, like adding it to cranberry juice or club soda, or adding some mint and ginger ale. Oh, there's tons of options. But this traditional recipe here, it has no heat, just acids.